Well, thanks a lot. I appreciate the invitation, and I'm always delighted to be speaking somewhere where I'm not jet lagged. I haven't traveled for 12 hours to get there, get off the plane, give a talk, and then get on the plane back to head somewhere else in the world. So I'm really delighted to be sharing with you what I've learned in the past 10 years about molecular diagnostics, biomarkers, and cancer. And so this title that was given to me is kind of like ambitious, the future of cancer biomarkers, what's in the pipeline. And so it kind of like stimulated me not to give the same old talk, but to go back and start a kind of like a brand new talk. So I'm inaugurating this talk here about the future of cancer biomarkers, and more importantly about what we have learned in the past 10 years. And I hope that maybe this would be of some use to you. And so being the head of the program in molecular diagnostics at the Hutch, then obviously it gives me a lot of thinking to do about what exactly do we want to accomplish in molecular diagnostics, in cancer molecular diagnostics. And so overall, there are a lot of opportunities to do things in molecular diagnostics, beginning with risk assessment, trying to figure out if we could develop diagnostics that would tell us who is at risk for developing this or that type of cancer. And beyond that, once we identify the subjects who are at risk, then can we develop diagnostics to be able to find cancer at an early stage when it's curable? And if we do find it in early stage, then what can we do in molecular diagnostics to classify that tumor molecularly so we could administer the best treatment that would be available in a personalized fashion, and then use molecular diagnostics or biomarkers to be able to monitor response to treatment, regression and progression type of thing. So this is incredibly ambitious, and a lot of effort is going into each one of those components. And I would say hundreds of millions of dollars are going into investigators' labs to try to see what we could accomplish at each one of those levels. And so given that I'm supposed here to share with you where we are heading and what we have accomplished, I could say a few words about risk assessment. There's been tremendous interest in the past decade in the post-genome era in looking at genome-wide association studies and see if we could identify subjects that are at risk for this or that type of cancer. And so, again, what have we learned out of this? And so very recently, a review, a meta-analysis came out in JNCI about what we have learned from genome-wide association studies for cancer. And this is the conclusion of the study. Although GWAS studies have identified several genetic loci, particularly for breast, prostate, and colon cancer, the explanatory power of these loci to predict individual cancer risk is very limited. And so, although there was a lot of excitement, or there's still excitement about GWAS studies, not much actionable has come out of that. And so, where do we go from here with respect to risk assessment? Well, it seems to me that the field is still wide open to try to figure out what sort of molecular approaches can we apply so that we could define risk beyond GWAS studies. There's some interest and excitement about moving away from GWAS and doing sequencing to stay in the genomic domain type of thing. But it seems to me that there's a lot more that could be done with respect to risk assessment outside of genomic analysis. And most of my talk is really not on risk assessment, so I would like to move on from this to talk about the main topic for my presentation here, which is what have we learned about classifying tumors in a molecular fashion. I'll say a few words toward the end about early detection, particularly for lung cancer. And by the way, most of my talk is going to be around lung cancer. I'll say a few things about colon cancer from a molecular classification point of view, but most of my talk is around molecular classification of lung cancer. And so in this post-genome era, if you want to start a new project or develop a career or want to embark on a a study to see what you could come up with in terms of molecular profiling that would inform, that would classify the tumor and inform about therapy. Well, you can be logical about it. And what does that mean? Being logical means that we have already established that there are a lot of genomic alterations in cancer cells, from mutations, methylation changes, amplification, deletions, rearrangements, what have you, that have a lot of bearing on whether or not a tumor is going to respond to this or that type of treatment, or to try to understand tumor behavior. But what we have learned is that what's going on in the cancer cell is not enough to understand tumor behavior or even to classify tumors. There's, on the other hand, the tumor microenvironment with infiltrating cells of different types, stroma, cytokines, chemokines, growth factors that play a very important role in tumor behavior. And if you want to classify molecularly that particular tumor, then that should be included in the classification scheme. And between what's going on in the cancer cell, what's going on in the microenvironment, then that determines the expression profile that you could analyze at, say, the RNA level and develop a classifier on that basis. And beyond RNA, which is really not very functional, then you could also do classifications based on the proteomic profile, which is more functional, the metabolomic profile. So you have a lot of different tools that you could use to be able to classify tumors, and with the purpose of deciding on therapeutics. And so there's been a lot of excitement in the past 10 years about doing this type of omic or global molecular profiling to classify tumors and predict different things: survival, outcome, response to treatment, and what have you. And so beyond all of those factors, there's also different components coming from the host, whether they metabolize, they don't metabolize. And so trying to develop a picture of then predicting response to treatment, among other, really has to include different components in this entire scheme. And so back 10 years or so ago, with the kind of beginning proliferation of omic type of profiling, particularly with DNA microarray at the transcriptomic level, then the current NCI director at that time, Rick Klausner, came up with this Director's Challenge, and some of you may be familiar with that program, challenging the community to develop molecular tools to classify tumors and predict response. And so I was at Michigan at that time, and so put together a program with multiple investigators to do that kind of molecular profiling for colon, lung, and ovarian cancer. And so the rationale for being so ambitious, wanting to do profiling on 3 cancers, was that it could very well be that there are pathways that operate in those 3 epithelial type of tumors, and learning something from one could be perhaps applicable to others. And it would be nice to see how the profiles from one cancer type to another then match or differ. And so there was a lot of excitement at that time with this kind of molecular profiling, and kind of like the joke was that within a few years, we're going to put pathologists out of business. We're going to have all those molecular profiles. We'll take that piece of tumor, we'll do this to it, and then we put it in the machine, and out comes the profile, how you should treat the patient, and what have you. And so there was a lot of excitement. And so out of that work that we were doing, which was transcriptomic, genomic, and proteomic, came out this first paper that we published in Nature Medicine. And so that would have been 2002, work that was done, say, 10 years ago, whereby we did transcriptomic profiling on lung adenocarcinomas, early stage. And lo and behold, we were able to come up with a 50-gene classifier that predicted survival in early stage lung cancer. And so this paper, which was published now 8 years ago, has had more than 1,000 citations. And so it was somewhat of a landmark in the field for a simple reason. That was one of the very first studies where we did molecular profiling on tumors that came from Michigan. And then we tested the 50-gene panel on tumors that came from Harvard. And lo and behold, then we were able to validate our classifier with an independent set of tumors. And so we're very excited about this. And this is what how the classifier performed in terms of predicting survival between the high-risk group versus the low-risk group based on this 50 gene index. And so it's not very dramatic, but at least the fact that it validated an independent set generated a lot of enthusiasm. And so there's been a lot of work using transcriptomic profiling by other groups also looking at lung cancer. And the sequel to this particular study was for the Director's Challenge program to do a second generation study whereby we compared different signatures that were available at the time and demonstrated that indeed we could validate some of those signatures in a blinded fashion in an independent set. And so the new validation panel included risk classifiers based on gene expression, but also included some of the clinical characteristics that were available. And so the upshot of this was that indeed we were able to come up with a classification scheme that predicts survival in early stage lung cancer based on gene expression profiling. So we were very happy. And what happened next? Well, this guy at the NCI, Richard Simon, a statistician, and maybe some of you might know him, then decided to take a closer look at the data that were published in different studies of gene expression profiles in lung cancer. And so the conclusion of his it wasn't a meta-analysis, it was a scrutinizing of the data that was published. And based on the scrutiny that he and his associate did, based on this review, we found little evidence that any of the reported gene expression signatures are ready for clinical application. Wow, I mean, a shocker. $100 million or more went into all of that work done not just by us, but everybody else. And at the end, he said, no good. And to make things even worse, he said that we also found serious problems in the design and analysis of many of the studies. And so dozens of studies that were done, according to Rich Simon, are really not worth a whole lot. And so what is the basis for his evaluation? How could he do this to all of us? And so basically he tried to apply objective criteria. He looked at the study design that was reported to see how appropriate it was and whether or not they did statistical validation in independent data sets. We did that. And whether the results were presented in an unbiased fashion, and whether there's any indication of the utility of whatever it is that the investigators came up with. And so what does he mean by that? Well, he said, for example, there are NCC guidelines for basically determining risk factors for lung cancer at the time of diagnosis. And so one of them is, well, was there any residual disease after surgery? And so looking at all those dozen plus papers that were published to see if they took into account in their design and the analysis of the data whether or not there was residual disease, well, information was lacking. And so by his definition, if you don't even know in the patients that you're trying to classify molecularly whether or not or who had residual disease or who did not, how can you come up with a classifier? And so this is legitimate. He had a whole laundry list of kind of complaints about all of those studies. Another one was to the effect that okay, in your study, you looked at 100 patients, 50 patients, 200 patients, but were those consecutive patients that were seen at the institution where you did the study, or is it pick and choose? And if it's pick and choose, then how do you know that those cases were representative of the general population of patients with the type of presentation that you are reporting on? And so he went on and on type of thing, but it made for an interesting reading. And more recently, in June, he came up with a review in Nature Review Clinical Oncology about what should physicians look for in evaluating prognostic gene expression signatures. And if you're interested in this field, I think this would be kind of a very interesting reading. Now, okay, he is like a lot of statisticians, very critical and doom and gloom type of thing. Is there anything that he could be excited about? And the answer is yes. He was excited about one study. It wasn't in the lung field. It was in gene expression signatures for breast cancer. And what study was he excited about? Well, the studies that led to Oncotype DX that I'm sure you're all familiar with. And he said, this is the prototype of how you should do your studies. First, find an interesting, important clinical application, and then let that guide your studies. Beyond that, whatever assay you come up with, it's not enough to come up with the assay you have, and being in lab medicine here, I'm sure you appreciate that. You have to demonstrate that the assay that you come up with is reliable and robust, that it could be implemented outside of the study where it was done. And then you have to be able to do validation on a very large separate group of patients, and Oncotype DX did that. And basically also beneficial if the assay that you're going to develop out of your study is one that is easy to implement, and if it could be applied to FFPE, that would be pretty good. And beyond that, then you should have a prospective study to demonstrate utility. And Oncotype DX is doing all of that. So that's the only one that he could cite that met his criteria. Now, what Rich Simon doesn't say in his review is that, well, Oncotype DX, as good as it is, or as limited the scope of that particular diagnostic test might be, has cost $100 million to get to the point that it's at today. So how could we, as investigators, then with measly little funding on an investigator by investigator basis or on a study by study basis, come up with $100 million to be able to develop this test for lung cancer or pancreatic cancer, colon cancer, what have you? We cannot do that. And so going over this 10-year history of hundreds of millions of dollars going to investigators' labs to do DNA microarrays or gene expression studies and what have you, and at the end, somebody with a critical mind like Rich Simon saying none of it is any good, is a kind of like an interesting situation to be in. What should we do about it? Should we quit doing our own research? Should we let companies like Genomic Health then be the ones to chaperone all of the diagnostics that we need to develop? This is a situation that we are in at the present time that you might have just as good an answer to as I might. And so one thing that Rich Simon left out from his analysis was that basically all of his considerations really were statistical. He wanted to have something that's statistically sound. So where I personally would differ from him is in relation to needing something not just statistically sound, but something that makes sense. And so we cannot just rely on statistics like he seems to do. So what does that mean? And I'll illustrate this with a parallel study that we did at the time we did the gene expression study. And so we were interested in doing omics profiling for lung cancer to predict survival. So one of the omics that we used was proteomics in parallel with what we did with transcriptomics. And so the proteomics was on the same set of tumors. And so we pulled out 20 lung adenocarcinomas, and we did their proteomic profiling. We were able to quantify 682 proteins and ask in a training and testing fashion, is there a subset of proteins that would predict survival in early stage lung cancer? And the answer was yes. And this is basically the curves that we got out of a 20 protein classifier for the same set of tumors that we did gene expression studies on. And so again, it was perhaps a generic set of 20 proteins, like we had a generic set of 50 genes with gene expression profiling. But what was interesting is that when we combined the gene expression data and the proteomic data and asked, are there any pathways that light up as being associated with poor outcome? And the answer was yes. What was interesting is that in the glycolytic pathway, just about every component there, either at the RNA level or at the protein level, then lit up as being associated, increased levels being associated with poor outcome. And so having a picture like this to me is just as satisfying, if not more, than simple statistics, because now we can relate to the data. We can relate to the findings, namely that overexpression of the glycolytic pathway, perhaps due to hypoxia, due to HIF1 alpha being upregulated, is associated with poor outcome and is indicative of aggressive disease. So now we can test this hypothesis in and by itself. And so the point I'm trying to make is with this types of omics approach, it's just not enough to rely on statistics. You have to have somewhat of a mechanistic understanding of why this list of 20, 30, 50, 100 genes or proteins or whatever it is seems to correlate with the feature that you are doing your study on. So, but the major lesson out of this, it seems to me, for all of us, and I strongly agree with Rich Simon and others on this, is for this type of profiling, it cannot be just an academic exercise. You can't say, well, let's look at 10 tumors of this and 10 tumors of that. Let's compare them and see what we come up with. And so if you're really intent on developing a clinical application out of this, then that's where you have to start. Define what your clinical application is, and that should affect your experimental design. And so based on what the specifics of the clinical application is, then you would carefully choose the patients, the subjects that would be in your case arm. You would carefully choose the subjects that would be in your control arm, and then do your initial discovery, and then a blinded validation in a completely independent set. Now, if you think that you're not going to be able to do that, then the lesson in the past 10 years is maybe you should not be starting this line of work, because it's unlikely, whatever finding you come up with, unless you have a roadmap to get you to the clinical application, 99% of the time, you're wasting your time. Okay, moving on from gene expression analysis to what is driving us crazy lately, namely wanting to find mutations in cancer. And so we're interested in finding driver mutations in cancer with the notion that maybe for this particular driver mutation, we could come up with a drug so that we could do targeted therapy and personalized medicine based on what mutations are occurring in this subject's tumor. And so in relation to lung cancer, a major study was published a couple of years ago in Nature looking at 623 genes that were rounded up because we knew already about their relevance to cancer, or that they had some mutations already described in lung cancer. And so those were analyzed as part of the TCGA program, looking to see what percentage of patients have mutations in any one of those 623 genes. And so this is the list that was that came out of that study of genes with mutations that were seen in tumors compared to normal DNA. And so it was kind of interesting because at least we have a comprehensive profile here of a very large number of genes on the one hand. On the other, very few of the mutations that were identified were really novel mutations. And basically, from a discovery point of view, it was more of a tabulation of what we already knew as opposed to a discovery of new mutations. Perhaps with the exception of NF1 as being mutated in lung cancer, just about all of the other genes where mutations were found, we already knew prior to this study that there were mutations in those genes in lung cancer. And so that was 2 years ago, but more recently, the number of genes with mutations that have been identified has increased, and there are some new mutations that we did not know about 2 or 3 or 4 years ago in lung cancer. And so this is the list of current mutations that have been identified as likely to be driver mutations in lung cancer. And what's interesting, and when you look at the whole chart, the whole pie of what a percentage of patients with lung cancer do we know as having a driver mutation, you'd see that for most patients, except for that pink slice there, already we know have a mutation. And a lot of those mutations, interestingly, a few of those mutations we already have targeted therapy against. And so those would include, obviously, EGFR, BRAF, and MLA-ALK. And I know that in lab medicine here, you already have then tests available for those particular driver mutations. Now, what we have also learned is that those mutations are not randomly distributed in the lung cancer population, and this is really typified by EGFR and KRAS that seem to be mutually exclusive, with the addition that EGFR mutations seem to occur in non-smokers or never smokers, whereas KRAS mutations seem to occur in smokers. So there seems to be some pattern in terms of the distribution of those mutations in lung cancer. Now, from a lab medicine context, the fact that we're seeing mutations there is not the end of the story, because those mutations could have been discovered using a particular platform or a particular technology, and it does raise the issue of, okay, how are we going to transform that into something that would be applicable in the clinic? And this is basically illustrated with the ALK mutation here, the ALK fusion gene, whereby there are many different ways that you could pick it up. You could pick it up by in situ hybridization, by PCR, by sequencing, or by immunochemistry. And so which one do you choose? So there's going to be somewhat of a chaotic situation in relation to trying to figure out how to implement a test for something that just got discovered. And so at some point, there's need to be a standardization of the methodology. And without that, I think it would be very difficult to make sense of different studies that have been reported. And I think in relation to this particular fusion protein, the use of immunochemistry seems to be on the upswing. Now, I would like to go through this particular case studies of dilemmas that we have. And the dilemma concerns EGFR targeted therapy and the choice of companion diagnostic to guide which patients then should receive EGFR therapy. So I don't know if this has been the subject of grand rounds before, but this is a story, a saga, and I call it a saga because it keeps changing all the time. And so I'm going to tell you perhaps the latest, and maybe 2 months from now there will be a different story about companion diagnostics for EGFR targeted therapy. So We have known about EGFR and its potential in lung cancer and other cancers for quite some time. We know that it's a major cell surface receptor, oncogene, that drives a number of signaling pathways. We have known that for quite some time, and this was the basis for wanting to develop anti-EGFR therapy for a long time. And obviously, more recently, in the past few years, we have learned that the occurrence of mutation in EGFR then identifies a subset of subjects that particularly benefit from EGFR tyrosine kinase inhibitor type of treatment. Now, from the point of view of trying to figure out how to predict response to EGFR therapy, then you have to know something about what EGFR does. These are all the players in the EGFR signaling pathway. And any one of them can mess up then what EGFR does or can do. And so trying to come up with diagnostics when the picture is so complex obviously is rather challenging. And so I've circled here two components. They are major and they are relevant to the story. One is PI3 kinase and the other one is RAS. And those have been, a lot of attention has been paid to them because obviously of mutations that occur in those particular components. Now, in relation to who is likely to benefit from EGFR-directed therapy, so we have known for some time that EGFR copy number and protein level predict survival benefit in patients treated with EGFR inhibitors. And more recently, we have learned that mutations in EGFR, particularly exon 19 deletion and other types of mutations, then make that tumor more sensitive. On the other hand, the occurrence of MET amplification as well as this other mutation at 790 then correlate with shorter time to progression in patients who are sensitive to activating to EGFR inhibitors. But that's really a small component in this saga. Now, what has emerged in a big way is the fact that a key element in response or lack of response is the occurrence of KRAS mutations. And in a meta-analysis that was published a couple of years ago, a review of 17 studies of 1,008 patients, 165 of them had mutated KRAS, then the presence of KRAS mutation was significantly associated with no response to TKI. And so this has emerged as a major indicator of whether or not a patient should go on EGFR inhibitor or not. A KRAS mutation means no response. And so, okay, given that information, how are we going to approach management of patients? Now, remember, on the previous slide, I showed how KRAS mutation is predominant among smokers, and EGFR mutation is predominant among never smokers, and in particular, never smoker Asians, and in particular, never smoker Asian women. And so do we need to get all of this information in terms of EGFR mutation, KRAS mutation, to decide on treatment, or should we look at the subject characteristic and decide on therapy without getting mutation status determined? And so a major study was published by Mock in Hong Kong with the Asian consortium that he has put together and addressing this particular issue. And so basically, the study involved non or light smokers with advanced lung adenocarcinoma, and they were randomized to whether or not they would get gefitinib, an EGFR inhibitor, versus the standard carboplatin plus paclitaxel. And these are the findings that came out of that study. So on the right upper hand corner, then you can see the impact of mutation in EGFR on the response to gefitinib versus carboplatin plus paclitaxel. And it's quite clear that the presence of an EGFR mutation then is highly beneficial in terms of treatment with gefitinib. On the other hand, if you look at panel C, which is the left lower corner, then patients who were mutation negative for EGFR actually did a lot worse on gefitinib, and they would have benefited from being on carboplatin plus paclitaxel. So it became very clear from that study that, well, you cannot embark, and the conclusions were that the presence in the tumor of a mutation in the EGFR gene is a very strong predictor of a better outcome with gefitinib. So it's not enough to have patient characteristics, but it would be extremely relevant to be able to determine EGFR gene status before embarking on gefitinib therapy. Okay, well, that means that we have to access tumors. And given the notion that there could be additional mutations that make somebody resistant, could we find other ways of predicting a response to EGFR therapy without doing molecular analysis on the tumor? So a number of studies have been published on serum predictors of response to TK inhibitors. MUC1 was published in 2007 as a reliable predictor of response to EGFR inhibitors in patients with bronchoalveolar carcinoma. Elevated serum level of sialylated glycoprotein KL6 predicted poor prognosis in patients with non-small cell lung cancer treated with gefitinib. Serum CEA level was found to be associated with EGFR mutation. Maybe it could be a substitute for that. And increased levels of EGFR, circulating EGFR levels itself was associated with response to TKI inhibitors. So there could be some blood-based diagnostics. But none of those really had much traction, as much as the paper that was published in JNCI with David Carbone from Vanderbilt and some of his collaborators. And so what they did there is take serum at the time of diagnosis and do mass spectrometry profiling. It's not the mass spectrometry that we do, that I'm going to tell you about later, but it's a simple mass spectrometry. They have been obsessed with simple so that it would be something that can be implemented in the clinic. So whereby they take the serum, they put it on a plate, they fire the laser, and they pick up flying objects, namely peaks, and try to determine profiles that way. But what was interesting and so that approach is really low sensitivity, plus the problem that you get peaks, but you cannot identify what those peaks represent by way of peptides and proteins. But nevertheless, they did this training testing approach, and they had a training set of 139 patients and then 2 test cohorts, and they were able to demonstrate that an algorithm based on 8 mass peaks then could predict response to EGFR inhibitors. So they got very excited about this, and I think they are pursuing this in a company mode at the present time. But in terms of the statistic, then this could be a type of study that meets some statistical criteria. On the other hand, remember the mechanistic plausibility. How can you assign plausibility when you don't even know what those peaks are? And so I remain somewhat suspicious until somebody tells me this is what this peak represents, and this is where it's coming from, and this is how it's related to response to TKI inhibitors. Without that, I remain somewhat skeptical. So is that the end of the story? The fact that we know that RAS is a bad predictor of EGFR response, and that EGFR mutation is a good predictor of response to EGFR therapy. Well, okay, I'll show you some more data. And I'm going to shift a little bit from lung cancer to colon cancer. And so there's something to be learned about KRAS, EGFR, and targeted therapy for colon cancer in advanced stage metastatic disease. And so quite a few, and so this relates to the Amgen inhibitor, the monoclonal antibody inhibitor, and how they got to get approval from the FDA to market that particular drug. And so the background for it was that we have known through quite a few studies that the presence of mutant KRAS correlates with poor prognosis. We have known that for quite some time in colorectal cancer. But beyond that, then it became clear that patients in early trials of the Amgen drug, those that had KRAS mutation, then did not respond to the drug. And so with that background, then in their phase 3 study that they designed, they included in their design looking for KRAS mutation to see what impact it had on the response to the drug. And so they did things in a meticulous way. They didn't just grab whatever assay for KRAS. They demonstrated reliability, reproducibility, as well as sensitivity in the company that they contracted KRAS assay to. And so they had evidence that the test is very sensitive. And even when there's 1% of mutant DNA in a background of wild-type genomic DNA in the tumor, they could still pick up KRAS mutation. And so the test that they used, and I'll get to that aspect in a minute, is one that would pick up 7 somatic mutations in KRAS in codons 12 and 13. And so when they finished this phase 3 study, then it became very clear that you have, like you see on the left side, then patients with advanced stage colorectal cancer that had wild-type KRAS then benefited from the treatment, and patients that had mutant KRAS did not benefit at all. So now KRAS becomes a companion diagnostic for the use of this particular inhibitor in the context of advanced stage colorectal cancer. Now, when you look at all the studies that have been published looking at the impact of KRAS mutation and response to this particular antibody, drug, then you could see that a certain percentage of patients with wild-type KRAS responded, and 0% of patients with mutant KRAS responded. Does KRAS explain response or lack of response? Well, not completely. And so what we have learned in the context of colorectal cancer is that there are a number of other mutations, like BRAF, like PI3 kinase mutation, that account for lack of response among patients who had wild-type KRAS. And so if that is the case, then wanting to determine who benefits from a TK inhibitor for EGFR, then it's not enough to rely on KRAS testing, but you have to round up additional suspects there before you initiate treatment. And so the plot thickens. It's not enough to determine KRAS status before you try EGFR inhibitor therapy. You have to basically round up the additional suspects and determine whether or not they are wild type or not. And the plot is even thicker than this with respect to KRAS. So the notion that has been ingrained is KRAS mutation is bad for EGFR therapy. And there was this guy, Bardelli, in Milan, who had a few cases with KRAS mutation that responded to treatment. And he was puzzled by it. How could it be that KRAS is so bad and I have patients with KRAS mutation that responded? And he would make that point in meetings and nobody would pay attention to him. And so he did a study. And this study got published very recently in October, at the end of October, a month ago, in JAMA, whereby he found that not all KRAS mutations are created equally. And so those patients that responded to the EGFR therapy, in fact, turned out to have mutation in codon 13, and the ones that were resistant had mutations in codon 12. And so this became this was a very important finding, pointing attention to the fact that you can't kind of consider all KRAS mutations as being equal, but there's a lot of minute details that have to be paid attention to. In this case, looking at specific mutations and asking the question, does this mutation impact resistance, or is it not relevant in the context of treatment response or lack of response? And so again, the story gets more and more complicated with respect to wanting to develop companion diagnostics on the one hand, and on the other, trying to understand why this subject is responsive to this treatment or otherwise resistant. Okay, so where do we go from here? Now we have BRAF, we have ALK, we have KRAS, EGFR mutation, and many additional mutations that need to be profiled and ascertained. And so what are we to do from a molecular diagnostics or a lab medicine point of view? Well, a recent editorial in JCO, very recent, came to this conclusion. Within the present era of personalized medicine, it's expected that this was a debate around KRAS versus EGFR therapy, that it's expected that colorectal cancer patients will have a full molecular profile. It's not enough to do this mutation or that mutation. You have to do a full molecular profile, as well as the possibility of repeat biopsies to see if the profile has changed over a period of time. And therefore, research must focus not only on developing new markers, but also on validating the testing that you would need to do, the testing methods, before you apply this in the clinic. Are we going to do a molecular profile on every patient that walks in before we administer treatment? Well, this seems to be the recommendation here. And in fact, some people are already doing that. At the present time, just a few days ago, this paper came in JCO. I don't know if you have seen it, by Dan Van Hoff in Arizona, a pilot study using molecular profiling of patients' tumors to find potential targets and select treatments for their refractory cancer. So in a series of patients, 86 patients, they attempted to do this gene expression molecular profiling, looking at a whole gene expression pattern to see if there is some indication of a driver pathway that might be suggestive of this treatment working better than that treatment. So he labeled this as a pilot study. Otherwise, you cannot get away with it unless it's a pilot study. And I'm sure he got all the human use approval that you need. But the point of it was that he demonstrated some merit in doing this type of profiling to guide the treatment. Obviously, it's not a brand new patient that you do this on. It's a patient that has failed whatever the standard treatment was that this patient was on. And they were able to demonstrate that there's some benefit in doing this molecular profiling. This just came out in JCO, and it was accompanied by an editorial by Dorosho from the NCI. So in his editorial, he was obviously extremely cautious about what this new approach implies and how we should be very careful before we embark on something like this. But to some extent, this is kind of like embryonic at the present time. But I would predict that in the next few years, we're going to see more and more of this heavy-duty molecular profiling, whether it's genomic, transcriptomic, proteomic, to be able to decide on the best therapy, at least in the context of patients that failed on standard therapy. Let me skip a few of those slides here and just say a few words about how we are approaching. We are doing the genomic sequencing. We are doing the transcriptomic profiling. And we're also doing proteomic profiling. And what we've been interested in doing the proteomic profiling is in coming up with blood-based diagnostics. And so the type of work that we do would be very similar to deep sequencing to come up with driver mutations or novel biomarkers at the genomic level. And so if you want to discover markers in the blood, you have to keep in mind or take into account the fact that there's a huge dynamic range of abundance of proteins in plasma. Trying to discover that one or a dozen or half a dozen markers that inform about lung cancer or colon cancer is extremely challenging. For every one of the potential markers like PSA, for every one molecule of those, you have hundreds of millions of more abundant molecules that are going to stand in the way of your identifying the low abundance potential biomarkers. And so the way we have approached this is in a kind of heavy duty fashion, whereby we take cancer patients' blood, we take control blood, we get rid of the abundant proteins like albumin, immunoglobulins, and we concentrate the remainder of the plasma mass, and we label it with isotopes so that we can distinguish case from control after we mix the two together. We do very extensive fractionation so that we could end up with discrete fractions, each with a limited repertoire of proteins, and then we do high resolution mass spectrometry on all of this. And when you do all of that out of one comparison of case with control, you end up with 200 gigabytes of data. Each column represents one of those fractions, and you have hundreds of proteins that you have profiled. And if you do a series of this, then you begin to see patterns of this protein being upregulated in this cancer compared to that cancer or compared to control. And you can get down to the depth where we can see things like EGFR in a mass spec fashion and contrast that with just having mass peaks. We don't know what they are in this other study that I described earlier. So we're using this very rigorous, in-depth proteomic profiling to come up with markers for common cancers. At the present time, through profiling lung, colon, pancreas, ovarian, and breast, we've been able to identify more than 6,000 unique proteins, each coming from a different gene, all in blood. And we have developed a database for those proteins across a very large number of subjects and cases that we have analyzed. So think about it. A third of the human genome is represented by protein in plasma that we have quantified and we have profiled in different individuals. And so out of that, what we are hoping is to come up with potentially risk factors, risk markers for the common cancers, as well as markers for early diagnosis. And so I'm running out of time here. I'm going to skip the rest. I'm going to go to the conclusion here and with the acknowledgement. This is work that started out at Michigan, like I said, 10 years ago. It involved a lot of people doing genomic, transcriptomic, proteomic studies, statistics, and mass spectrometry. So I would let the credit roll while I answer any questions that you have. And thank you very much.
Questions. Well, there's a question back there.
Yeah. So I haven't read the article by the statistician that you mentioned at the beginning, but one of the interesting things that struck me.
It's great reading. I recommend it.
Well, one of the things that struck me though is that the criteria that he proposes seems to have been met by, as far as I can tell, none of the actual standard diagnostic techniques that we actually use today, like histopathology, even histochemistry, at the time of their implementation.
That's true.
So then getting to your last slide or a later slide where you mentioned that simply validating the techniques that we actually are using today, is that maybe where we should be spending more of our focusing?
Well, I would say you have to do both. One, validating the techniques that we're using at the present time for sure, and the best case for that is PSA. The data from different assays for PSA were completely different from each other until there was some standardization, and now you can relate findings no matter who the vendor is for the PSA assay that you're using. So this is obviously quite essential to do, but I think his point was going to standardizing, even going up to the discovery part and applying rigorous criteria to your data. Even at that point in time, in the roadmap toward the clinical application is going to be very important.
I was curious about how Simon's work has impacted the thinking at the NIH of how they should fund this type of work. Are they going towards mega centers?
Well, I really brought up this particular study for exactly that reason. And so there's this big conundrum for NIH. Do we fund research that's investigator-driven, or do we fund research that is consortia, multidisciplinary, multi-investigator, big team type of projects? And so there are the proponents who say you cannot touch single investigator research portfolio. This is absolutely essential. And they make the point that all the major discoveries have come out of single investigators. So this is kind of like you cannot touch that. But if that's the case, how are we going to do the examples of genomic health, $100 million study that it takes to do something from beginning to clinical application? And so this is a dilemma, and I don't think that NIH has addressed that in a meaningful fashion. And so right now we are kind of stuck.
I mean, just talking about single investigator is a bit of a misnomer based on your credits rolling there, right? That there's a lot of collaboration going on, you know, even between single investigators now. So.
Right, but I can tell you that we absolutely have no means to drive an application all the way to the point of taking it to the FDA. So there's this issue of where would academics stop and then turn over things to somebody else. And now how many, and when you look at the cost of Oncotype DX, $100 million. How many companies are there? No matter how exciting you're finding at the research lab as a single investigator or as an academic, how many are willing to cough up $100 million to say, okay, I'm going to take this to the clinic tomorrow? Not very many.